You can. Can you make it a full screen? Oh, there we go. Yeah.
Yep. Is that okay?
Yes, looking great. Fantastic. Thank you. All right, everybody. Welcome to the August Seminar for Own Your Future Mentorship Program. As some of you know, and may be joining us, our mentorship program runs, the seminars run every month. Sometimes they break in seminars for conferences or other competing medical imaging events. So we're happy to have Dr. Sola Adeleke today doing our entrepreneurship, the mentorship seminar cycle between clinical, which is heavily on radiology, physics, a lot of the research side, and then entrepreneurship. I really want to stress that entrepreneurship because it cuts across both radiology, basic science, especially in Africa, where we have funding limitations and often funding coming from external. We have to learn really how to run the labs as small businesses. So this entrepreneurship seminars are really great for every discipline. And we're happy to have Dr. Sola Adeleke today, bringing back the entrepreneurship seminar for us. For those that are joining for the first time, the seminars typically go for about 40 minutes to 45 minutes of presentation. And then we kind of go into this Q&A section. The Q&A section is more so directed at you understanding what the seminar was. So there's going to be a lot of echo back and forth between the presenter and yourself. So we encourage you to engage. You can engage by chat. When we have poll questions running, we're going to ask you to go and participate in the poll, because that way we are able to engage, get the feedback, and also make this whole session lively. This is not a webinar. This is a meeting format. So I'm going to ask you to mute yourself when you're not speaking. When we get to that engagement Q&A networking kind of session, then you can unmute yourself and we'll be able to have really good meeting. So without further ado, I'm going to introduce Dr. Sola Adeleke, who is a dear friend of mine and a very supportive advocate for diagnostic imaging, its application in both clinical setting and in research. And also essentially a very strong advocate of making sure that this imaging modalities are inclusive and accessible that everybody in the world, including our colleagues in Africa, are able to use it. So we're really happy that he's here to give us his time. He is the founder and chief medical officer of Qnetics, which he'll be talking about today. Dr. Adeleke is also a practicing oncologist, and this Qnetics came from essentially him looking at the current approach to clinical care in cancer, especially looking at immunotherapy and how AI can be used to enable treatment outcomes and being able to actually do better prediction. He will not want to admit this, but he's actually a really good MR person. He did his PhD in image-derived, so MRI-derived biomarkers for prediction in prostate cancer. We've done work together on cardiac MRI. So I think from an imaging perspective, we have a very fantastic speaker. And from a fellow Nigerian to you, Dr. Adeleke, I forgot the Yoruba term. I practiced it, but I forgot it. I was going to say thank you, Yoruba, but I forgot. So I'm going to pass it back to you. Thank you so much for being here.
Thanks. You do not think the word we're looking for is Eshe or Adupe?
Yes, Adupe. That's what I was looking for. Yeah, it slipped my mind.
Yeah, and I see myself as a pretend radiologist. I'm just pretending to be an MR expert, you do not. But thank you for the vote of confidence. Means a lot. Hello, everyone, and thank you for logging in. Apologies for the initial IT issues. I think we're now here. And I'm excited to be here, and I look forward to sharing the next half an hour or 40 minutes with us and telling us about Qnetics and what I do and what the company does and how this links in with some of my passion in terms of getting access to cancer care, not just in the West, but also in lower middle income countries. So Qnetics was founded by me in 2018, and we're a cancer AI company. And what we do is to improve patients' response to treatment, particularly immunotherapy. I'm going to tell us more about that in a few slides. So it was one of my associates that put this here. I don't like to put all of my stuff here, but I think just for context, it's probably good to let people know that I was born and bred in Lagos. I did my medical training there. I finished 2007. I was at Lagos State University College of Medicine, Keja. Before then, I actually did a physics degree at University of Lagos. So I was Unilocked from 1998 to 2001. So Greater Akokite, I was there, and then went to the other side of the state to the State University to do my medical training. And I started residency in radiotherapy or oncology at Luth in Lagos. I did that till about 2009 or early 2010. So I was there for about 15 months, 18 months as a registrar, as a resident. And that's how I started my journey into oncology. And it was quite an exciting journey, even though we had limited resources there. It really stimulated me to think about, thinking outside the box to solve very pragmatic, very practical problems in terms of access to care. So I moved to the UK. I got a scholarship to study a master's in oncology. I went to University of Nottingham in 2010. And before then, actually, I applied for the Apprentice UK. And I got to the second stage of that, which I'm proud of. I didn't get to the TV stage. So it was quite exciting. We didn't get to meet Lord Sugar, who is like the Trump of the UK. Well, not as sarcastic as as we were talking about. So it was quite an exciting journey for me to understand, what it takes, how to have the guts really to stand out amongst the crowd. And just go back a little bit. So when I was in medical school, I'm sure we do know this. If you trained in Nigeria, there are a lot of strikes, and lecturer strikes, student strikes, and the program was meant to be 6 years. So I entered 2000, I finished 2007. I didn't fail. It was just like because of the extension of our training, because of dispute over pay, over quality of training, over access to resources. So during one of those strikes, I went to learn a trade. So I went to learn fashion design. I don't think I've shared this with Duna before. So I went there. So it was a 3-month strike. I was like apprentice at a fashion design shop in Mushin area of Lagos. And the reason why I did that was as a medical student, we wear white coats, and our white coat is shorter than the doctor's coat, the qualified doctor's coat. But what tends to happen is that there is just a box standard coat that everybody wears, whether you're big, small, whether short, tall, it doesn't matter. So we're just wearing baggy coats. And I said, we can be more trendy, we can do better than this. So I went to this, it was just serendipitous. I went to this apprenticeship. I learned how to measure. I learned how to do some sewing. I'm not very good at it, but I did know how to measure and know how to fit people. So when we went back to school, I set up this business where I was doing bespoke house coats or doctor's coat for my class. So I started with my class, about 60 people. Within 2 months, all of them got their lab coats sewn by me. Then the second year got to know about it. And eventually, within a year, I was a sole fashion designer for the whole of my medical school. That was about 500 or 600 people. And that money was actually very important, very crucial for me to, go through medical training. And it was excellent. In fact, towards the end of my medical training, someone wanted to buy off that business from me and carried on, and even had pharma or small pharma company trying to let me make their lab coats for their lab. So it was quite an exciting. So I caught my entrepreneurial tooth from that journey, and ever since it's been fantastic. So I did an MBA at University of Lagos, and the MBA was an eye opener for me, because going from A levels to medical training, you're thinking a particular way, think like a scientist, thinking about physics, biology, chemistry. You don't tend to see the other side of the world about economics, about marketing, about management. So I did an MBA. It was an executive MBA. I had to pray and beg my dad to pay for that. He wasn't ready to pay for it because he was already paying for medical training. and he couldn't understand why she also paid for a very expensive MBA. It was a lot of lobbying from uncles, from sisters, how the African thing works. They come and, cajole him. So, he's got this passion for it, give him a chance. And I think it was quite expensive. So we're talking about 2003. It was 250,000 naira per year. I think if you look at inflation now and all of those things, it's probably going to be about 3, 4 thousand pounds, in today's money for you to pay for that. So, and it was a part-time MBA for 2 years. And I was still in medical school. So I finished lectures in medical school around 5. The lectures for the MBA start around 6:30. So, and it lasts to about 8:30. So I was doing 2 schooling, really, medical training and MBA at the same time. And I also happened to be the class rep for my MBA. So I was leading an MBA class, all the managerial stuff, and liaising with the lecturers, arranging tests and assessment or assignments, and case studies, all of those things. But what that gave me actually gave me access to some of the brightest people in the area of business in Lagos. Because some of the best people in banking, in IT, in oil and gas came to that MBA school. And many of them are still my friends till today. In fact, many of them have been ministers. One of them is still a minister now in Nigeria. He's had his own oil company. And many of them are directors of banks now in very big banks in Nigeria. And many of them are doing really well even abroad. In fact, one of them was my best man. He used to be a director at Procter and Gamble. He paid for our honeymoon in Ghana. And with his network in Ghana, I was able to meet one of the ministers in Ghana. So that's all the story in terms of how I started off in Lagos. Then I came to the UK, did my medical training, did my MSc in oncology. Then I went on to do my PLAT exams to practice in the UK. I did that. Then I wasn't very sure whether I was going to stay in the UK. I did um, the USMLE exams to practice in the US. I did the 3 steps, step 1, CK and CS, very gruesome exams. It took me about 2 years to do because, each step you have to practice, prepare for about 6 months. And I was working full time as a doctor as well. So I did that. But family happened. I decided to stay in the UK and went into training, did my membership at Royal College of Physicians to become a physician. Then I've done my fellowship at Royal College of Radiologists to become an oncologist. It's a bit funny the way it's a membership board certification exams. And I also did a PhD in MRI, like what Eugenia said. So that's my journey. Sorry, it's a long story, but I just felt I should let you know. So going on to kinetics. So this is a team. So it's quite a diverse team. So we have people who are in IT, people who are in head of commercial. We have Professor of Immunotherapy who is working with us as a collaborator on some funded projects with his university. We have a Professor of Molecular Oncology. We have Professor of Machine Learning. We have AI scientists, oncologists, cancer geneticists. So, and we have the FCCA, Chartered Accountant, with our CFO. So this is like half of the team. We have a few other people who are on this card on this slide. And we also have a small team in Toronto. We're just setting up, and we're going to be setting up a small team as well in Boston within the next few months. So what we do is, like I said, we're using novel AI tools to predict whether a patient is going to respond to immunotherapy. And we need to understand the biology of the disease. We need to understand the clinical management of those patients with cancer. I also need to understand the AI bit of things. And that's why it makes it quite a complex problem, because very rarely you have someone who understands biology, computer science, and also oncology or medicine. It's very hard. So we had to bring a team together. It took me about 2 years to bring together that team. And it's just very difficult. So I'm going to play a video now. I'm going to just come out of the sharing and share my other screen so you can actually see the video I'm going to show you now, just to give you a better view of what we do. Let me see if that works. Stop sharing, and I'll share again now. So just give me.
Ongoing battle against cancer, immunotherapy emerges as a powerful treatment. While immunotherapy holds promise in cancer treatment, it's expensive, can sometimes be ineffective, and often triggers undesired side effects in 55% of patients, some of which can be fatal. Until now, there hasn't been a reliable method to predict in advance whether a patient will respond positively or negatively to treatment.
I don't think we can see the video. I can only see.
Curenetics solves this problem by utilizing the power of artificial intelligence to analyze vast libraries of anonymized data from reliable sources such as the NHS. Through the analysis and combination of genomic, histopathological, and clinical data sets, we can better understand and predict whether a patient will respond positively or negatively to treatment. Curenetics' easy-to-use software can be integrated into existing systems with ease, allowing for connection to hospital records, so that with a click of a button, our patented AI system will deliver personalized results in a matter of seconds. This will not only help doctors offer patients the very best treatment strategies tailored on an individual level, but will also help to save hospitals time and money on ineffective treatment. So if you're ready to shape the future of cancer care and save the healthcare industry time, money, and lives, book a demo of the Curenetics platform today at curenetics.io.
Thank you for that. I'm just going to go back to my other slide now. Are we taking questions now or waiting till when I finish? I think.
Oh, we can. I can run a poll if you want to, but just one thing, so that you know, I don't know if that was a video or supposed to be an audio only, but we didn't see any of the video, but I think the message was clear. It's just that I wanted it to
Sorry
be.
about that. I thought the video was sharing.
Yeah, I think that's fine. I think the message was clear in there. So whenever I'm ready to run a poll, I could launch one.
Yeah, should we do maybe 1 or 2 polls now and see what people think?
Sure. I think I have up to question number, what am I now? 15 ready. So if you want to do any one of them, just let me know which number and I will launch it.
First 2 questions. Should we do the first 2?
First 2? Okay, here we go. Just give me 1 second. So let's see how this works. Let's go back to poll and start with the first one. 1 second. And make sure this is there, and let's just waiting for it to launch. Okay, so I have the first poll launched. I don't know if you can see it. Sola, can you see it? Can everybody else see it?
Yes.
Okay, so the question is, what is the primary objective of Cunetics AI algorithms? Is it to develop new cancer drugs? Develop new cancer drugs? I said that twice. Okay, so yes, so develop new cancer drugs, provide general health advice, conduct clinical trials for new medications. And there's one more question, one more option that was missing, which is predict treatment outcomes based on molecular data. So which one is it?
I think that's a giveaway.
Yes.
Yeah, the last one's the giveaway.
Well, we are not conducting trial. It's the one that we didn't mention, which is not on the list. That's the right answer.
Which is to predict treatment outcomes based on molecular data.
Yes.
So can you talk a little bit about that? How you predict treatment outcomes based on molecular data?
Yes. So this is what tends to happen. So when a patient is due to have treatment for cancer and they're eligible for immunotherapy, they have their tissue sample, which is a biopsy, and they also do some genomic profiling on them. So they do what we call programmed dead cell ligand protein assay, PD-L1 protein assay, which is expressed on cancer cells. On the surface is a receptor protein on cancer cells. And what that protein does, it actually dampens the effect of T cells. And if you remember from our biology, T cells are like the army of the body. They go around looking for foreign bodies or for infectious foreign bodies to kill off or to engulf and destroy them. So when the cancer cell expresses PD-L1, the T cells will sense that, they would latch onto that. And by doing so, they become ineffective. So they cannot kill that cancer cell. They see the cancer cell as one of them, not a foreign body. And when that happens, then the cancer keeps growing because it just keeps proliferating. So we now know that PD-L1 on its own is not very good at telling whether a patient is going to respond to treatment, because normally what we expect or what we see is that the more than PD-L1 expression on the cancer cells, the higher the chance that they will dampen the immune system. And hence, if you take that breaks away, you might have to get a better response to treatment if you have the right treatment for them. But that doesn't always happen. So the drugs we use to bind to the PD-L1 receptor are called immunotherapy. They unleashes the body's immune system against cancer. So, and a host of them, we have nivolumab, atezolizumab, pembrolizumab, durvalumab, and cemiplimab, about 10 of them, or just about. And they are used in different cancer types. But what we do see is that only about 15 to 30% of patients respond to treatment if we use that approach. In fact, many people that we give immunotherapy to do not respond. Majority of them do not respond. And about over half of them will have some side effect of toxicity. And only about 20% of them will have sustained response, if they do respond at all, in terms of having response that continues for about a year or two beyond their treatment course. So we now think that if you look beyond the genomics, which we currently do, if you look at the imaging, so look at CT scan, your MRI, or scan histopathology slide, H&E stain slides, and look at clinical data from the patient, maybe you can get some insights or deeper insight into why some people respond to treatment and why some people don't. And we've been able to show that by using that combination of genomics, which we already do have, imaging, we are always scan patient with CT every 3 months or every 6 months, or some of them might need a PET scan or MRI. And we combine that with the information about the patient, a past medical history, or past surgical history, or comorbidities, or list of medications. All of these have an impact on how you respond to treatment. And that's what we do. We combine all this multimodal data, and our algorithm is able to analyze those complex data sets, imaging or clinical or genomic. And we can actually tell on a very personalized level whether a patient is going to respond to treatment or not.
Fantastic. So do we go on to the next question?
Yes.
Okay, so let's just, I fixed the problem now. So I'm going to launch poll number 2. What is one of the significant problems with the current standards of care, predictive testing for immunotherapy? Is it because it's too expensive, it's too inexpensive, has low predictive accuracy, it's always accurate, has no side effects? 6 out of 16 people think it's B.
Yeah.
Any other takers? What do you think, Sola?
I think they've nailed this. I think they've been listening. That's great. I'm not boring them off. So it's true. So it's because the current way of testing or predicting response is very poor. And like I said, it's about 30% in majority of cancer types. And that's just what I've just explained. So, and we know that one of the reasons why we're doing this is because we know over 70 million people are living with cancer globally. And many of them will be eligible for immunotherapy because the FDA approvals or EMA approvals for immunotherapy for either first line or curative or palliative intent is getting longer because the drug it could be very efficacious if you use it well for the right people, but it's expensive. Many of them are still within patents. In the UK, we talk about £8,000 per patient per year for immunotherapy per person. In the US, it's over $100,000. In fact, that's even an underestimate. We're talking about $150,000, $200,000. And you have a situation where we're giving the patient a drug, which you don't know whether it's going to work. And if you're talking about cancer, you don't have time to waste, really. You don't have 6 months to wait in some cancer. You want to get it on right away, and you want to get it right the first time. So that's one of the reasons that saving cost, saving lives, as well as improving, the chances of survival for many patients by prolonging a life in doing so, and also avoiding side effects if not going to be the right treatment for you. So I've explained this. I'm not going to belabor it. So our models is able to predict accuracy by over 80%. So if you remember, we're talking about 30% with the current standard, in some cases 20%. We're now getting, in fact, we're getting 80, 85% now in terms of predicting accuracy to treatment. And so that is one of our products. Should we ask some more questions about immunotherapy before I go about our next product that we have within Kinetics? Or the
Yeah, do you know which question number that could be? Is it number 3,
maybe,
number 4, number
maybe
5?
the next 3 questions.
The next 3 ones? Okay.
Yep.
So let me just pull that up. That would be number 3. Poll number 3 coming right up. Okay. What percentage of patients typically have a sustained response to immunotherapy? Is it 10 to 15%, 20 to 25, 15 to 20%, 30 to 35%? That's a Jeopardy if you're wondering what I'm humming. One thing I didn't have like Sola is multiple talents. I did actually, but that was in junior high, fashion designing, but that was like in high school for 2 summers. So we did make our high school uniform as well.
Wow.
But I wasn't that good. So when I was coming to Canada at 15 years old, when you're making that trip back, I don't know, because I grew up at UNN, a lot of kids like myself, we all moved back to where we were born. Everybody tends to want to dress as English people they see on TV. So I sat down and made a Nigerian outfit because I wanted to make it. So that saved my butt because when I got to Amsterdam, huge airport, connecting as a 15-year-old, I was so lost. So I stood out in my African garb. Look like an 8-year-old walking around in African garb. And so that's how I got people to help me because I looked so like I stood out. Yeah, but that's how, as far as my fashion sense went. I did not turn it into a company.
Well done for that.
But see, 60% of people think it's 15 to 20%. What do you think?
I think so. The numbers we've given is between 15 to 30, so 15 to 20, just about right. So well done for everybody that participated in the poll. Should we go to the next question?
Yes. Okay, let's go back to number 4. How much can be wasted per patient per year in the healthcare system due to ineffective immunotherapy? This is in pounds, so it looks like a lot of money. 85,000 pounds. What is that in dollars? That's like almost 100,000 Canadian dollars. That's a lot of money. 100K, 70K, 50K, which one do we think it is? They all think it is around that sweet number of 85K. Is C always the right answer? What do you think, Sola?
Yes, that's correct. So we put it about 85,000 pounds. So which is spot on for
patient
most people.
per year.
Yeah. So it's not cheap.
Okay. And I think this is one of the motivations of what AI can do in terms of trying to limit that. Okay. Do we go on to the 5th one or?
Yes, let's do one more. Then we'll tell you about what we do.
What specific types of data does Kinetics AI platform analyze to predict immunotherapy outcomes? Is it genomic only, genomic imaging, CT/MRI, imaging only, clinical trial data? And I guess clinical trial data also includes the non-imaging blood-based biomarkers, right? I think they'll think it's genomic. It's all just throw everything kitchen table at it. Genomic imaging and clinical data.
Absolutely. So well done, everyone. That's the right answer. So B, and so it's a multimodal approach that we're using to solve that complex. Genomic will not give you the kind of accuracy we get, obviously. So we're gonna do the remaining 10 questions once I've given some more thought about key myths, and by the end we'll do the remaining questions. And if there's any Q&A from the audience, we also take that. So QMate, so you might be wondering, we're talking about immunotherapy and we're now talking about COVID PCR. What's that? So 2 years ago, we started working with Premier University of London. So it's one of the London-based universities. And we worked with a wet lab group there. And it's a cancer molecular biology lab. And what this lab has been doing over the last 10 years, the professor in charge of that lab is to develop a PCR-based test even before COVID came on board, a PCR-based test which is able to diagnose cancer and specifically oral cancer. So I'm going to tell you how this is important. So oral cancer, even though we don't see it quite often, it could be quite a debilitating disease, disfiguring disease, because you have to hack into people's palate, into mandible, into their jaw to get a cancer out. Some of them lose the ability to speak or eat properly or dry mouth. So many horrible things happen to them. So the incidence is expected to double by 2035. And the conventional way of biopsying people or making a diagnosis of oral cancer is to take a chunk of tissue, which is about 50 millimeters, usually by an oral surgeon or dental surgeon in a secondary care setting in the UK. And you might need suturing with local anesthesia. And you'd have to wait for about 2 weeks to get a result back from the pathology lab. And you have to make an appointment with your doctor to reveal the result of the appointment of the test to you. So there's a time of anxiety that you have to wait, not knowing whether you got cancer or not. And for many people, even if they don't have cancer at that time, many people with high risk, like smoking, alcohol, chewing tobacco, or HPV infection, all the other things that might increase your risk of developing oral cancer, they might have to do this every 6 months or even every year. So, and it's a very traumatic. We spoke to some patient advocacy group. They said that the biopsy itself, not the treatment for the cancer, is what gives them the PTSD, because you have to do this regularly, where you have to take chunks out of your mouth, and you have to go through local anesthesia, not proper anesthesia, for you to be able to go through that. So what this lab at Quimeri has done is that they now say, can we reduce the footprint of biopsy in this patient? Can we actually take a lesser sample and even be as accurate or more accurate than the standard of care? So what they've done is that just by looking at a half grain of rice, so like 1 millimeter cube of tissue, they're able to test within the clinical environment using a PCR machine. And within 90 minutes, they'll tell you whether I've got oral cancer or not. And that's phenomenal. And the way they do this is that they identified 16 gene panel within the cancer, which they've patented in the UK and the US, and building an algorithm, a statistical algorithm to analyze the gene expression profile across the 16 genes. They're able to tell whether the patient has oral cancer or not. And where we come in is that the accuracy of the test using just statistics is about 80–85%. But when we went to start working with them as a team, as a company, we're now able to get over 90%, 95% accuracy by using our AI platform to analyze this gene profile on the strain. And we got a grant from NIHR last year to do an excellent validation work in China and in India. So all the work that's been done so far has been in the UK. But using our model, we're able to show that actually it's reproducible, it's repeatable in a different patient population, in a different healthcare system in 2 countries. So on the back of that now, we now got regulatory approval in the UK. We got a UKC approval, and we're just about setting up for commercialization of this technology. So this might actually be a good thing in Nigeria or developing countries where they don't have access to secondary care that can do this diagnosis for them. But many centers now have a COVID PCR machine because of COVID. And many of those machines are not being fully utilized because the incidence of COVID is lesser, or the virulence of the virus is lesser, and people are not dying from it. So we're now thinking that, can we actually utilize that same equipment to improve the diagnosis of oral cancer. And we're even doing that in other cancer types, not just oral cancer. We're looking at squamous cell cancer, cervical cancer, and other cancer. And this is just a demo of how the test works. So just a tiny bit of tissue, 1 millimeter cube or 2 millimeter cube. We put it in this preservative reagents that we've done. Then we use this mRNA Dynabits technology to extract the mRNA. Then we put a 16 gene panel we've extracted on the well. Then we put it on a PCR machine, and we have a readout, which is now sent to our AI model within the same 90 minutes to get the results back. And this is phenomenal. This is one of the samples that's been taken. You can see very tiny tissue, just little tissue. Very, there is no, you don't even need it. You don't even need a suturing when you've done this test in patients who are high risk of oral cancer. And this is a kit that we're going to be selling in the next year or so, or less than that. And we got CE marking for that technology. So should we do some more poll? Some of the poll will still be on the therapy, just to see whether we're keeping our attention going.
Which numbers should we
from
do?
5 to 10. Should we do 5 to 10?
We did 5, so I think you mean from 6.
6 to 10.
Yep, here we go. How does the AI platform's ability to analyze gene expression patterns contribute to predicting treatment response? Is it by identifying specific mutations linked to drug resistance, by correlating gene expression patterns with potential immunotherapy success, by determining the most common side effects of treatment, or by predicting overall survival rate of patients? Why do you think they all think it's B?
Because it's the right answer.
Okay, so linking gene expression to potential immunotherapy success.
Yeah, because even though we're not looking at genomics alone, but on this poll, that's the most accurate answer. So we're looking at gene expression profile with imaging features and clinical data. We combine that on our AI platform to predict response.
That's phenomenal. Okay, I'm going to move to the next one.
Yes.
Okay, it's poll number 7. Which imaging modalities are integrated into Kinetics AI platform for treatment response analysis? Is it X-ray, ultrasound, CT, MRI, PET and SPECT? That would be nice. Mammogram and bone scans. Most think it's CT and MRI.
I think they're right.
Why not PET? Because on nuclear medicine, on molecular medicine, no, imaging, I should say, because, cancer has that molecular signatures that sometimes you kind of miss with MR and CT. Why not PET and SPECT? Will PET and SPECT be integrated later on?
It will be eventually. And the reason why we're going with CT and MRI is because that's the scans we do regularly in patients. They might have a PET scan at diagnosis, but they don't have it every 3 months like CT scan. Well, you're right. We're going to include that eventually.
Okay, fantastic. Number 8. What is the major advantage of using AI to predict immunotherapy outcomes compared to conventional methods? It eliminates the need for any clinical data, offers a non-invasive predictive method with over 80% accuracy, replace the need for imaging altogether. I don't know. It guarantees 100% accuracy in predicting treatment response. My imaging bias, C is just like, as an imager,
Yeah.
Hard to replace imaging.
So a lot of people are signing up, which is great. So we have 11, and they're all right. So it offers non-invasive predictive method with over 80% accuracy. So even if you don't have genomic data, we've run some experiments where we just use clinical data alone and imaging alone, and we're still able to predict very highly the accuracy of the patient responding to the therapy. We don't always have to have genomics. I know in low resource setting where they don't always do genomics, we can still do CT or clinical data to still predict response to treatment. So well done.
Number 9. Kinetics platform is designed to optimize which aspects of cancer care or cancer treatment, dose levels of chemotherapy, selection of targeted therapies, success rate, cost savings, and decision making at the molecular level, development of new surgical techniques. I think it should be all of the above, but I think that's just a process still waiting to happen.
Yeah, yeah.
But for now, what can the platform do?
So I think the
That's a problem. Everybody thinks it can do everything or should do everything. So what at this point, what does it do, Sola? Where are we at?
so we at sea for now. We're making decisions about at the molecular level. You're right. We're not there. We are not focusing on targeted therapies just yet, and we're not developing surgical techniques. Definitely not. So it's not a D. Maybe in future, and we're not looking at those levels of chemo. So for now, we're focusing on immunotherapy. Thank you for all the people that voted on this. That think we should be able to do everything. But
Well, it looks like you're getting there. Yeah.
so
One more, and then we go
one
to the
more, then
next.
we go to the next. Yeah.
Yeah. So, is the platform designed to optimize which aspects of cancer? Oh, I think, hold on one second. This may have been a repeat. It's probably a repeat. Let me just stop that. I may have repeated the question. Okay, let me just go to this one here and launch that one. What percentage of patients typically experience a toxic or negative? I don't think you've covered this, but I will go ahead and put it. What percentage of patients typically experience toxic or negative effects from immunotherapy according to the data presented? I mean, these are all high. 25% is still a large number, even if that's on the low end.
It seems you're influencing some people. Once you said 25%, some people
Oh
jump.
yeah, I'm just saying, even as low as 25, that's a lot of. I'm surprised to see these numbers. That's a lot of, and that's also because it could be expected, right? You're dealing with immunobiology, and people are different, and there's obviously diversity issues with some of the way this therapies are designed, and so I could see those being higher than 25. But what are we at in terms of percentage of patients that experience toxic negative effects from immunotherapy?
So on my screen, 55% of patients have been reported to have toxicity from immunotherapy, which is quite high. People wouldn't expect that for a licensed class of drugs, but it does happen. But the thing is, the toxicity could be very mild, just like a dermatitis rash, which you can manage with topical steroids, to having colitis or hepatitis or pneumonitis or thyroiditis or all those itis really, which could learn during intensive care. So it's a wide spectrum, but data have shown that over 50% of people will have some side effect, and that's the reason why we think that if it's not the right drug for you, we can avoid all those rubbish side effects, which might kill some people. People die sometimes from immunotherapy, so it's not trivial at all. So yeah, I think that was a tough one, and C and D was just about close. I think D will be the right answer based on data that we have. Well done, everybody, for giving it a go. And yeah, so I'm going to be moving towards the end of my presentation, and then after the presentation, I have the remaining 4 or 5 questions to go. So the oral cancer test, we have regulatory approval in the UK. We're going through UK EU approval. So as a company, if anybody on this call are thinking about, oh, I want to start my own thing, I want to start my own company, I do encourage you to do so, because there's a lot of health challenges or resource challenges in low and middle income countries, in Nigeria, in Africa, that many people are already solving. And fintech is still the big thing in Nigeria, solving access to banking services, and people are now doing a lot of health tech. Well, I might be wrong. Many of the health tech solution I see is around telemedicine in Nigeria. That's what many people do. But I think we can do more than that. We can solve our own problems. Can you solve malaria? Do we have to wait for Belinda Gates to do that for us? It's not bad. We're getting funding from it. It's amazing that they're putting their hard-earned resources to help people that don't have that resource. But we understand our problems more than anybody else. Can we find a way to solve problems like typhoid or multi-drug resistant TB or diarrheal diseases in children? There are some challenges there that people can solve really that can extend life. You don't need the grants, or you don't need funding from IMF or the World Bank to solve. You can solve this yourself, and people will buy the solution. So if you're thinking about starting your own thing, whatever you do, you need money. So when I started off, I put all my savings into this, everything I've got, I put it into this business. That was about 3 years ago. Everything. And I even borrowed money at the beginning just to pay salaries at the beginning. So it's not trivial. Now we've raised over $1 million. We're just about raising 5 million over the next 6 months. And looking back now, I think it was some of the best decisions I've ever made. Some of the money I borrowed, I'm still paying it every month from my own salary to where I got the money from. So it's a very challenging journey, and it will deny you of your free time, your evenings, your weekends. You don't know how I talk about this when we're writing grants for research. So we're doing that, and you also have to think about paying salaries the following month for people that have mortgages, that have family, and you have to think about that as a CEO. So then you have to juggle. So I got a full-time academic career job, clinical career job. I work in the NHS. And so most of the things I do is either before 9 a.m. when I have my meetings, or after 5 o'clock. Or I have many meetings on weekends as well to catch up with the team and to troubleshoot and do so. Almost all of my free time goes into academia, clinical practice, and research. So just to let you know that if you want to do the same, because I couldn't afford to come out of clinical work because that's where I pay my mortgage from. And we don't have enough money to pay me and pay the rest of the team at the beginning. Now we're getting better now in terms of financial status. So when you've now developed the product, you also need to think about regulatory. So whatever product you develop, I think NAFDAC will be there. If you're going abroad, FDA. If you're going to Europe, you have EU, EMA. So you always have, and those things take time. So we're going through EU approval now. We've got a UK, we're going through EU and FDA. FDA is faster. We think we'll get a FDA before the EU one. And it takes about 12 to 18 months, and it's not cheap. So when you're developing your prototype, when you're developing your technology, always think about all of these things. And you need a lot of people to support you. And the critical thing about being a founder is about your personality more than your ingenuity, actually. Your ability to convince people to believe in your dream and believe that this is something worth their time, worth doing, and also committing their time and effort into it, and also convincing people that they should invest in you and invest in this solution for the betterment of people with cancer and beyond. So you have to put yourself out there. I go to networking events, startup events, coming out from clinic, long clinic at 5:30. I go down central London, still mingle with people till about 8, 9 p.m. I have to be in the clinic in the morning at 8 o'clock or 8:30. And I have to do that because that's how you get people. You meet people, you know that person can get you in front of someone in FDA, or that person can get in someone in front of a hospital director, or can get you funding from a VC. And that's how you have to just keep doing that every time. So now we know, I think this is the last 2 slides or so. We have a lot of challenges, goes without saying, in our home countries. And as scientists, as clinicians, we're clever enough to be able to solve that problem, even though we don't have the support of our governments or our system for this to happen. There are now venture capital in Africa, in Nigeria. In fact, Nigeria, Lagos is a startup capital of Africa now. And I think Joburg or Cape Town, then Nairobi as well. And Accra is not far behind in terms of doing a lot of this innovation. So if you're thinking about, you want to solve problems, because the government cannot solve all this problem. Even in the West, Now, the government doesn't solve all this problem. Some of this are left to the businesses to solve. Even in the US, they have all the money, even in China. So we, as clinicians, as scientists, as physicists, as radiographers, as radiologists, we know where the pain points are. We know where the problems are. If you think about it, if you need help, seek out help from people that have done it before. And that's how you have this forum. And that's how we're having this kind of mentorship, like Udina and the rest of the team has arranged so nicely. And, we can bounce ideas back. Or how can you solve this problem? Access. Low field MRI came out of, some of these challenges as well. People can access, 3T machines or 1.5T machines. And a company now is now making six formula Tesla machine, which is now being sold to Africa, and people in Uganda are now solving their own problem. We all know who Jonas is, and those are the kind of innovation we need, even though it's not commercialized yet. I can see that happening over the next 5 to 10 years. So we need to think about those problems as Africans. We don't have to wait for other people to come and solve this problem for us. And those are the kind of challenges you face: lack of skills, workforce shortages, or training, or cost. And they just have to keep going. It's not easy. It's really, really difficult to innovate, really difficult to become an entrepreneur. In fact, many businesses fail by the first, second year. We've been going for about 4 years now, 5 years now, and we're not even there yet. So it's going to be a high risk, high reward venture, but I think it's worth doing. And you might end up with some bit of debt at the end of it if you're lucky to fail. That's the part of the problem as well. So yes, so that is, I think, the end of my talk. Let's do some polls, then we take questions.
Yeah, no, this is fantastic. I mean, before we get into polls, I just have one thing I wanted to touch on, which is this, because looking at your at the company right now, and wasn't really designed to solve, like you mentioned, our African problem. It's designed to solve just a clinical healthcare problem across all settings. So from this idea of reverse innovation, this idea of trying to innovate in a resource constrained setting, and how that actually on its own, it actually confers some amount of advantage, in the sense that if you make it work in a low resource setting, it will almost work and easily translated in a high resource setting, like in the West. And also, if you design it for low resource setting or for African challenges, you're not able to address all the potential pain points, and you make this in a cost-effective way. So can you just add a little bit of thoughts to that, and then we can go on to the poll questions?
Yeah, thanks, Udina. So that's very important, because if you can strip it to the bare bones in Africa, all the remaining are going to be belt and braces really in the West. So if you can make it work in a very resilient system, like in Lagos or in Accra or in Nairobi, and is well tested there, there's a good chance it's going to work elsewhere. You might have to, pivot. You might have to adopt or adapt some of the few things that is not obvious in Africa to the different healthcare system. But in learnings from innovating in a very hard, resource poor environment will help you going forward. And just to tell you about immunotherapy, that the AI platform, so we're making a platform. So I haven't talked about the other program we're running. So we're building a cancer vaccine program where we're using AI platform to identify epitopes of T cells to identify more immunogenic cancer vaccines. We're doing that in the UK. That's a different program. We're going to have a postdoc starting in January for that. And we're also having a program where we identifying people that will develop stem cell transplant rejection. It's called graft versus host disease. We've already done that with academic. We've done some papers. We're now doing some national validation. So with the same AI platform, we patented. So how our technology applies to Africa is that we're looking for partners. If anybody on this call could be that partner or knows someone who could be that partner, we want to see whether we can use models to identify who is going to respond to chloroquine or artemisinin or antimalarials or not, or who is going to respond to typhoid treatment. There are different treatments. Many people are now resistant to typhoid antibiotics, or even sepsis that kills a lot of people, and we have limited access to very expensive antibiotics in Africa, or even multi-drug resistant HIV or TB that kills a lot of people in Africa. So we're looking to, I think, our platform with the right data and the right people to support us, we should be able to solve some, low resource setting problem by using the same technology we're using in the West.
No, that's really fantastic. And that's what I think the message here is, especially for the mentees in the audience, is that they should, I mean, it's not they shouldn't look to the West. I think they should look internal, because if you make it work in Africa, if you use that idea of reverse innovation, you are actually going to be a lot more successful, in my opinion, translating it to the West and making it a successful company because of that low cost way of looking at problem solving. Poll number 14, right? Get on to that one.
Yes.
Okay, so I think this is a question you had before the QMed. So this question is, how does Qnetics AI-driven approach potentially reduce healthcare costs associated with immunotherapy? Is it by eliminating the need for follow-up appointments, reducing the number of ineffective treatment administered, avoiding the use of immunotherapy altogether, minimizing the duration of hospital stays? Okay, ineffective treatment, reducing the number of ineffective treatments.
Yes.
Bringing that 55 number down. That's, yeah.
Yeah. I think that's the correct answer, B.
Right. So the goal is to bring that high number of failure rates, and that's okay. Which specific biomarker are likely being analyzed with Qnetics AI platform to predict immunotherapy outcomes? Is it hormonal levels, PDL1 expression and tumor mutation burden, glucose level, electrolyte balance or imbalance? I thought it could with a combination of both the tumor expression, tumor burden, information you get both from imaging and genetics, and as well as the hormonal levels. Hormonal levels, you don't consider that. For certain cancers, you might, right? But maybe not for the...
Yeah, for breast and prostate, we haven't done much work on that. You're right. We will do that for those kind of cancers. Or
cervical cancer and some other ones that may have.
That's right. Yes.
Yeah. Or thyroid.
Yeah. B, absolutely.
Okay. We're doing good for time. 16. How does Qnetics AI platform utilize clinical data in conjunction with genomic and imaging data to refine predictions? I like the idea of refining predictions with imaging, ignoring clinical data when genomic is available, integrating clinical history such as patient comorbidities, prior treatment response, contextualized genomic and imaging data set, focusing solely on imaging results, using clinical data only for initial screening purposes. Now, this is a tough one. I think the integration of, because you guys use it's a multimodal approach to integrating clinical history with morbidities and treatment response within the context of genome, which is where you're doing the refinement, right?
Yeah.
Yeah.
So
that is a great approach. And so do you have multiple different AI? So that's one thing with AI is there are so many methods for one problem. People do have, GON, CNN, VNet, UNet, everything just to segment the brain or everything to predict something. Why is there not a one-stop shop that could do exactly one AI model that could do the prediction, history? Do you have used multiple models to do this, or just one-stop shop model?
excellent question, Abdina. So for the genomics, we use, decision trees like random forest, XGBoost, for the imaging, you're right to use DNN, CNN, graph convolutional neural network, GCN as well. Or even we're now using transformers as well for images. And for clinical data, textual data, we use NLP. So we still have to bring all of and our proprietary models we develop in-house. We have to bring all of this, existing models and our own models together to solve this problem. So there isn't one single model yet or algorithm yet.
Right. Okay. And what are the ethical considerations associated with using AI to predict patient response to immunotherapy, privacy and data security, bias in training that could lead to unequal treatment outcomes or maybe even harmful treatment outcomes, informed consent, use, and all of the above? Do you agree it's all of the above?
Indeed, yes.
All of the above, indeed. Okay, and I'm just going to flip to QMIDS. What is QMIDS? Is it a new cancer treatment, a diagnostic test for oral cancer risk, an imaging technology, a surgical procedure? What is QMIDS? Yeah, I think they paid attention. This is a digital diagnostic test for oral cancer risk. Anything else you want to add to that? I think they're right on the ball. Right on the ball.
Spot on. Yeah.
Spot on. Okay. We have maybe 4 more, so we're doing good. How many patient markers does QMed assay measure to produce a malignancy index? 10 markers, 12, 16, 20. And this malignancy index is based on healthy versus patient across the spectrum.
Yeah.
And so you had some healthy patient, some healthy control data sets.
Yes, for the
Okay,
training.
and this is all from the NHS?
Yeah, so
or
mostly
from research?
From the NHS and also from abroad. So we've done it in Scandinavia, we've done it in India, we've done it in China as well.
Oh, fantastic. Combination of NHS, and the healthy controls came from NHS or from research?
From NHS too.
Oh, this is why NHS is great. This is why, public healthcare is great.
It's got its challenges, but it's got its own.
Oh yeah, that's something that's good for, I think.
So 16 is
besides
right.
The waiting, what is the significance of malignancy index generated by QMED's assay? Is it to determine the need for chemotherapy, provide scores between 0 and 20, which is likelihood of a lesion being malignant, measure the patient's overall health status, or replace any further diagnostic testing? Some think it's to provide a score of malignancy in terms of likelihood of it being malignant. And this will be very important when we move to other applications beyond breast. I would like to see how this works within a brain imaging context, and I think we're getting there in terms of having that rich data set. Brain tumors are very rare, so they're not as and that's one thing with small data sets. If this would segue into that a little bit, data sets. How big is big? How small is small? How much data sets do you need to be able to have this predictive malignancy index?
Yeah, so I'll answer that. So the answer here is B, so they're right. Likelihood is 0 to 20. So when I asked our data scientists and academic collaborators how much data, they said you can't really do sample size, really, calculation in AI, to be honest.
Yes, that's what they claim.
So you can't really do. So we have our own internal threshold that if you want to do any modeling, we need to have at least 250 to 300 at a minimum for some pilot data work. If you want to do some validation data work or large data work, we need at least 2,000 to 3,000. So the minimum we'll say is 250. Anything less than 200, you're going to be, getting some wild, biases, and it's not going to be able to give you some good inference from your results. So we don't really do anything less than that.
Okay, we got 3 more. What are the advantage of using QMed assay over conventional oral cancer detection methods? Less invasive, faster results, higher accuracy, more invasive, but quicker results, similar invasiveness and accuracy, but faster results, more invasive and slower, but highly accurate.
So
I would rather have less invasive. I don't like needles.
Yeah, so
less invasive, faster, and high accuracy.
That's correct. So that's A.
Fantastic. And we got, in what ways does the integration of AI enhance the predictive capability of QMID's assay? The platform analyzes the malignancy index in conjunction with patient data to refine risk, slowly provide a second opinion for scoring patient data. It has no role whatsoever in QMID's assay.
So That is right. A is right.
Essentially in that malignancy index. Yeah.
Yeah. Yeah.
Okay. We got a couple more and then we turn around. We have 10 minutes for questions. Which patient population? Oh, I didn't launch it first. Which patient population is particularly suitable for QMIDS assay? Patients or individuals with no history of oral issues, with non-healing mouth ulcers or suspicious oral growths lasting weeks or months, confirmed throat cancer, routine dental checkups. So when they start to have suspicion of ulcer or oral growth, that's when you think that it might be good to start your QMIDS assay, because then you can predict who might end up having that lesion or that ulcer, which one of them could actually become malignant.
Yeah. So the answer is B. So the next iteration of the KMIT is that it's going to be like a self-testing kit. People, smokers, or people with previous risk of cancer or alcoholics, they can actually do the test at home using a brush. So we're going to combine, rather than taking a biopsy with a tweezer, we have the brush biopsy now. You put it in the solution, we send it, just like you're doing pool test for bowel cancer. You can send it to the lab and we can get it results within a day or two. So that's going to be in another 1 or 2 years. That's what we're hoping to do.
Fantastic. All of those sewing and business training, good use. What challenges might arise in the widespread adoption of QMIDS assay and its associated AI platform in clinical practice? The challenges: resistance from healthcare providers due to complexity, high cost, integration of existing health IT system, all of them and more.
Yes, all of the above.
Resistance for healthcare. Can you talk about that? Is it because they are not aware of what is under the hood to know, how well this could, the risks involved? Or is it because it's a new technology and it's just something that they don't understand?
People are just averse to change. They're just resistant to change because, it's something new. They don't know how it works. We try to make it quite easy for them. So it's just about the anxiety of changing, your pathway to something different, and how would that impact the way of you doing your work. So it needs a lot of reorientation and hand holding and support, over a couple of weeks or months for people to get to use it.
Right. And I could see that for all of the AI applications as well. So I think the floor is yours. We have 8 minutes. We did good, even with all of the hiccups. This was fantastic. And if you have any questions for Dr. Adeleke, based on everything, not the sewing stuff, but on kinetics, this is your time. You can ask 2 ways: type in chat, or mute yourself, and then we will be able to take on the questions, or he'll be able to take on the questions. So there's a lot of residents here. And I think one question that if I were a resident in Nigeria, hearing this for the first time, with the current practice or training environment, AI is not part of the training yet. I don't know in radiology if it is. I doubt that it is. How did you start coming from an oncology, doing the residency in Nigeria, like you said? Where was that AI? What was the first time you learned AI, and what processes did you use? Was it online training? Were there very good training resources that you can encourage? Spark being one of them, but
Yeah, Spark is excellent. It's good you mentioned that. So I did a physics degree, like I said. So I already had some background in programming. I wasn't an excellent programmer, I must say, but I already understand the logic behind programming. So when I started my PhD, I had to use a lot of MATLAB as well, for analyzing some of my imaging data. And UCL was quite good. They had a 6 months boot camp where PhD students do analytical PhDs and some postdocs are giving this training for free. I think it's 2 days a week or 1 day a week for 6 months, where they'll train you on how to analyze genomic data using R MATLAB, and Python.
Mm.
And I found that was a turning point in my understanding of AI, really, even though I wasn't very heavy for AI. But the Python bit was really good for me in terms of how I got to know it. Then, when we started Kinetics, my understanding of AI was just like anybody else on this call. But the first grant we had was an EU grant to work with the university academic group, AI academic group, and we hired a fellow who is a data scientist. He's now our PhD student, he's finishing his PhD end of this year, and I was the secondary supervisor for the PhD. So every Friday we had a lab meeting where I was sitting down as a clinician, and the other supervisor was an AI professor, and we have the data scientist. So they're talking about AI every week, and I was just soaking everything in for about a year. So it was like an informal training for me, really. So I'm able to speak to my data scientists now, what they're doing. I'm able to critique them. Why are you using that model? Why are you using that algorithm? Why are you combining this? Or why can't you do early fusion or late fusion or mid fusion for your models? So that's how I picked it up. So going back to how in a certain Nigeria where there's limited access to AI, even radiologists in the UK, they don't have AI as part of their curriculum. I'm not sure about the US or Canada. So everybody is still not very trained yet in AI. But people go on courses. So I've got a medical student from UCH in Ibadan who approached me on LinkedIn. He was a fourth year medical student when he started working with us. And he did his online courses, lots of online courses. During COVID, he was also doing some online courses with a team where he was analyzing some clinical data from Kenya. And he came for online interview with us with our AI team, and it was brilliant. We gave him an internship with us for about 7 months. We're paying him in pounds, and it was based in Ibadan as a medical student. So I think he was living big life, really. So just to show you that the skills is really, really excellent. It's difficult, especially coming from a biology brain to a physics brain is difficult. But if you can take yourself through it and learn it, people will need that too, because not so many people understand medicine and understand AI.
Yep.
It's a very rare skill to have.
Right. I agree 100%. I see that a lot with my clinical colleagues. All right. So questions. People are wanting your LinkedIn link to connect with you. Good luck with that. When you share it, expect the connections. So they want you to share your LinkedIn connection. Any other questions, comments? This is your time. He's a busy guy, as you see. And one thing that Sola does well is when he gives you his time, you have his time. So this is his time now before he goes and does the 5 different things that he does: clinical research, clinician, entrepreneurship. Let's see what question. Oh, he put the LinkedIn. So this, I think the Q&A section did actually allow them to understand what kinetics is. I think for me, one thing I would ask is, when is this coming to Nigeria? When are you going to have your Lagos office? You have a Toronto office, and I'm putting you on the spot. But when are you going to have a Lagos office? And what would it take to have a Lagos office? And this is also for them to understand when they are thinking about setting this up in Nigeria. When, like, you talked about Lagos being the hub. When is that hub going to be? When is Kinetics going to be in that hub?
Yeah, thanks for doing that. So I keep thinking about that. So the 2 things we need is to have a clinical collaborator who can work with us. Then we need the funding that can support that kind of research, academic, or commercial collaboration. And we need to know the research questions to answer. Are we answering imaging questions? Are we answering, communicable disease questions? I'm interested in that because many people suffer from that, from malaria, from typhoid, from, all those diseases that, we take for granted in the West now. So if anybody think that they have the right, expertise, or they know people that have the right expertise, and also funding to be able to do that, because raising funds in the West, I don't think we'll be able to use that money to do R&D in Africa.
You're right.
It's difficult to justify that to our Western investors and say that.
It is.
There's a different class of VCs that do that, on the continent and elsewhere.
Right, right. And those investors always come with invisible strings attached. So I think, and this is one reason why the seminars are great, is that we need to start to raise our own capital, to do our own research, to do our own clinical translation, our own VC and the entrepreneurship companies. Money from the West always comes with strings attached. So this is fantastic. Sola, this is great to have you. Thank you so much for giving us your time. There are opportunities for internships. People are asking for that. If you want a PhD student, I think you've opened up the bag by talking about the student you had at UCH that you paid in pounds. So here comes the questions. I think for internship opportunities, I would say that you can go to Kinetics website. I'm just going to pop that in chat, and so maybe faster than I finding it. But I find it here. Kinetics.
Yeah.
You may find it if I do. There it is.
I'll just type it here.
Make sure it's the right one.
Yeah.
Is that the right one? I always have to go check. That's the logo. Yeah.
Yeah.
Got the logo here. Okay. So, oh, here you did it. Yeah. So I'll say that you go there and there is a careers. I'm just going to pop in the careers link here. And I think this is where you can find information about internships and studentships. And again, if you're resident, if you're senior, if you're junior consultant, and you have some ideas, there's some funding that you found, reach out to Sola. We'll be happy. He'll be happy to sit down and see how they could brainstorm around the grant to make it work. Okay, so I think we can call it a day here. We did good for time. We're right on time. So this is fantastic. Thank you so much, Sola, for your time. Safe travels when you come to Canada. We'll be going the opposite direction when you're here. But I'm sure that this is going to go places, and as I think we all here can say, as Nigerians, we're super proud of what you're doing. It's always good to see Africans doing great in the West, and I hope that this inspires everybody. I mean, I'm inspired just listening to you. I thought I was working a lot, but now you make me feel lazy. So, so this is very inspiring, and I'm hoping that this will actually get a lot of people following your path that you sort of blazed for us.
Thank you, Dina.
So thank you
so much.
Thank you all. Enjoy the rest of your week and your days. And how do you say bye in Yoruba? Oh my God.
Odabo.
Odabo. That's good night. Isn't that good night?
Odabo is bye. Odaro is good night.
Oh, but forget it. I love the above. Odaro, Odabo. Bye, everybody.
Bye. Thank you so much. Bye now. Have a nice